Hey guys, so this video covers the process of clonal selection, still part of our immune system PowerPoints. So our learning intention for this video is to understand how lymphocytes, which are T cells and B cells, are able to react to a wide range of pathogens and to know how the process of clonal selection occurs. So clonal selection is a theory which explains how our B and T cells can respond to a wide range of pathogens. We know that there are so many pathogens that our body's able to respond to, and we have to look at why that occurs. And what it does, it allows us to be a much more specific response to a particular pathogen. So how it works, so our B and T cells are developed in the bone marrow, and each B and T cell is made with different randomly generated receptors. And those receptors are particular to each antigen or pathogen that comes into our body. And as there are different millions and millions of T and B cells, that means there's a large number of receptors that could be made. So you can imagine here that let's say that these are 4 T cells that are being developed. One's been developed with a square-shaped receptor, a round, more triangular, and a more oval-shaped. So each of those receptors, they're almost predicting what are the possible things that come into our body, and they're making a wide range of, as I guess a prevention type thing for when those antigens enter our body. So when we come across a specific antigen, only the T and B cells with the specific receptor are activated. And that means that that specific cell is going to clone or replicate and make copies so they all respond. So if you look in this diagram, you can sort of see how this might be 4 different B cells, and they've all been made with different receptors. And only this B cell has the right receptor for the particular antigen, in this case, the red triangles. And when it becomes activated, it is going to clone and replicate many, many times. And we get a large number of cells that are being made specific to that one antigen our body's encountered. We don't need the other 3 at the moment, as we're facing a triangle antigen. These other 3 B cells won't actually work. And so there's no point spending energy replicating those cells over and over. We only want to replicate and clone the cells that we know can respond to the antigen at that time. So some more examples. So you might have T helper cells. They go through clonal selection. So here is a dendritic cell. It's doing antigen presentation, but only the specific T helper cell is going to be activated. So here, these are these little red semicircles. This T helper cell's got the right receptor, and it's going to then replicate and clone. There would be other T helper cells that don't have the right receptor, and they're not going to be activated or cloned. Here you can see a B cell has come across a pathogen, and again, it's the specific B cell. It activates through a T helper cell, and it's going to be then specific to that exact pathogen. It's going to clone and replicate and make many, many copies, okay? And they're going to produce the specific antibodies, which we'll get to in our next video, against that pathogen. And here the same again, this cell is displaying some sort of pathogen inside it, and only the specific T cytotoxic cells are going to respond, and they're going to replicate and make copies. So this is all an example of clonal selection. We have one cell that has the right receptor being activated and cloning and replicating many, many copies. And the cells that don't have the right receptors don't copy. Again, just more examples. You can see a specific pathogen binds to the specific T helper cell. That T helper cell is then going to activate specific plasma cells, which make specific antibodies. It's going to activate specific memory cells. And so you can see, again, these cells that don't have the right receptors aren't being cloned. And so they're not being selected. We're picking the one that has the right matching receptor. So what's the benefit? Well, being able to do that means that our B and T cells are able to react to a wide range of pathogens. If we make millions and millions of different receptors, it means that just by chance, when a pathogen comes into our body, we may have the right receptor to fight that pathogen off. It also means we save a large amount of energy. If we're only spending energy cloning cells that are required, it means we're saving all that energy rather than replicating and cloning all cells. And so it's much more specific. And the biggest one is that if we have memory cells developing and they're cloning, it means that we can have a much more specific long-term immunity against that particular pathogen. So all of these are highly beneficial to the immune system, makes it much more efficient and much more targeted. And that's why clonal selection is a really important part of adaptive immune system. So in review, our T and B cells, which are developed in the bone marrow, have a wide range of possible receptors, and only the cells with the specific receptors are activated when a pathogen exists. And they then replicate and proliferate, which means make many numbers, and create that specific immune response. And that's much more efficient than trying to replicate everything all at once. So I hope this video helped. Thanks, guys.